The OMIQ workflow begins with uploading data. Data files are chosen from your local computer. A key first step before analysis begins is to tag files with additional metadata. This can be done simply by copy-pasting a spreadsheet. Metadata can be categorical or continuous for different forms of statistical analysis or functionalities within OMIQ. File names can also be changed at the same time during this process. A similar process can be used to correct any conflicting channel names. Here you can see that I'm correcting a conflict between two batches of files. We can also change the display names of all the channels. Analysis in OMIQ is done by selecting tasks. Tasks are building blocks of individual functionality and operations that can be taken on data. The list of available tasks is growing constantly. Currently, it spans operations in pre-processing and gating, dimension reduction, clustering, trajectory analysis, differential statistical analysis, and various forms of visualization. OMIQ also supports custom tasks for enterprise clients. Custom tasks are unique visualizations or scripts written in any programming language and installed into OMIQ directly. They can accomplish any goal, such as customized PDF reporting, autogating or other forms of cellular classification, or other proprietary actions for your enterprise, such as sending analytical results to an ELN. If you do not have code to supply, but you have an idea of what you want, we can develop the custom tasks for you. Tasks are connected together to form workflows. This flexibility allows any data to be processed however it needs to. Each task has a dedicated page to configure it. In this compensation task, I have two groups, and I can edit the matrices and see plots update in real time. As workflows become more complex, their structure is critical to stay organized and maintain auditability and reproducibility. Note that workflows also get locked at certain critical points to ensure this. In this analysis, we were looking for differential cellular signatures in GVHD patients at 30 days and 90 days post-bone marrow transplantation. Our first step is to do data pre-processing. Next, we will use tsni-CUDA for dimension reduction. This will give us a good idea of the high-dimensional structure of our data in an easy-to-digest visual format. tsni-CUDA is a form of tsni that runs on GPU and is over 100 times faster than normal tsni. Next, we'll do clustering and metaclustering, followed by differential statistical analysis and different forms of visualization to help us interpret the results. We'll start by looking at our differential analysis results. Reviewing the results on a volcano plot shows us the clusters with the largest differential abundance change, as well as statistical significance corrected for multiple hypothesis testing. Because this result was discovered automatically, we would now try to figure out what these clusters are. Using a clustered heat map can help us understand the high-dimensional signature of each cluster and which clusters are most similar to each other. Here we can see the two metaclusters of interest, number 22 and 28, clustered near each other in the heat map, and both express CD19, CD45RA, and differ on some other markers. To continue to enrich our understanding of these data, we can use OMIQ's plotting capabilities to rapidly drill down and create whatever types of figures and plots that we want. Some tasks, such as FlowSum, may produce a graph as one of their results. If that's the case, we can open it up and analyze it in OMIQ's Graph Viewer. Here I'm activating metaclusters to show their location on the graph. Once graph nodes activate, so do the box plots, which show sample-level statistics and information. This significant metacluster, number 1, looks to have an extreme outlier in one of the samples, with the rest of the samples being fairly rare. Significant metacluster number 22, which is high on CD19, among other things, has a very clear and consistent differential abundance between groups. We can further analyze this graph by changing the channel or changing the file as desired. We can assign nodes, which are clusters, from the graph into metaclusters as we see fit. Alternatively, we can import predefined metaclusters onto the graph. After importing the metaclusters, they can be edited. This means combining them, splitting them, removing them, and otherwise redefining them. To really dig into our data, it's possible to have the graph and a figure open at the same time. My selections on the graph become selections at the single cell level within the figure. This strategy can also be reversed, where plots are gated in order to find out which nodes activate in the tree. This same technique can be applied for gating as well, where the plot on the left controls the updates to the plots on the right. In both cases demonstrated here, note that on the left we were looking at single files, but on the right, the plots are configured to overlay multiple files at the same time while also overlaying the filter coloring. If we instead want to rely on manual gates instead of clusters for our populations, it's easy to fork the workflow and then still plug in those gates to the same statistical techniques. Alternatively, we could fork the workflow to try a different form of clustering, either to corroborate our results or to get a different perspective on these data.